One. Hi everyone, my name is Nisha and I'm joined by Vicky, Maggie, and McKaylee for our Spring 2024 Functional Genomics poster presentation. Our title is Analyzing Amyloid Precursor Protein-Protein Interaction Pathways in Different Organisms and Susceptible Populations in Relation to Alzheimer's Disease. Alzheimer's disease is an interesting neurodegenerative condition as it impacts memory, thinking, and behavior. We specifically looked at the amyloid precursor protein, or APP gene, and its variant. For some background and introduction, Alzheimer's disease is the most common form of dementia in elderly adults. It is characterized by impaired memory and thinking abilities, and at the fundamental level, the accumulation of proteins called amyloid beta plaques and neurofibrillary tangles. The APP gene provides instructions for APP production, so mutations in this gene can contribute to disruption in neuronal communication, neurotoxicity, and inflammation. It is crucial to shed light on the impact of health disparities on certain racial and ethnic groups, as prior studies have found that African American and Hispanic populations are more susceptible to Alzheimer's disease.
To conduct our research, we came up with two main objectives. Our first objective is to examine diverse protein-to-protein interaction networks of APP across three distinct organisms. They are Homo sapiens, which are humans, Mus musculus, which is mice, and Drosophila melanogaster, which is the fruit fly. Our second objective is to investigate frequency distribution of the SNP associated with Alzheimer's disease to identify the risk profiles among various populations. We also came up with two hypotheses. Our first hypothesis is that the fruit fly will exhibit the most distinct protein-to-protein interaction pathways given its taxonomic classification as an insect, in contrast to humans and mice, both of which are mammals. Our second hypothesis is that African populations will be at a higher risk to develop Alzheimer's disease because they will be more likely to acquire the variant C allele in comparison to other populations.
To answer the first objective, we first used StringDB and searched the APP gene. We examined the protein interaction of humans, mice, and fruit flies. The parameters were set so that no more than 10 interactions were shown in the first shell and no interactions in the second shell in order to see the proteins directly associated with APP. To answer the second objective, we used the GWAS catalog to find the SNP associated with APP. This was searched in Ensembl to explore the population genetics of the SNP. The results display the string interaction of humans, mice, and fruit flies. The different colored lines represent known interactions, predicted interactions, and other interactions such as co-expressions and protein homology.
We also made a table to depict the string amyloid precursor protein-to-protein interactions of humans, mice, and fruit fly for biological processes, specifically in gene ontology. At the top of the table, we have the protein-to-protein pathway, and on the left-hand column, we have the pathway function and organism name. As you can see in our table, we have similar pathways for the human and mice. However, for the fruit fly, we can see that we have two separate pathways.
The population genetics show the proportion of the C and T allele of 5 different populations for the SNP variant. T is the ancestral allele and C is the variant, making Africans the least susceptible to acquiring the variant allele and East Asians the most likely.
The APP pathway function comparison supported our hypothesis, with the fruit flies only having two biological processes that were quite different from those of the mammals, and that the mammals were very similar looking at the 5 selected on the table. The allele frequency distribution gave us some surprising results with the African populations, with the higher proportions of the T ancestral allele compared to the other populations. The previous literature shows increased vulnerability for them as well, which would be interesting to look into further. The critical evaluation portion, we have strengths in the fact that we had good similarities and differences in our protein pathways, and they were consistent with our hypotheses, and that we had diverse representation in our research. A limitation is that we only have the biological processes for our gene ontology and not for molecular or cellular components. There is some ambiguity in the T and C allele representations, and then the only SNP variant we studied was the APP. The future direction is that we want to look into medicinal treatments for pathways involving APP interactions, and we also would like to look into susceptibility into further demographics such as sex, income, and age. The allele distribution demonstrates a trend contrasting that to the founder effect. Thank you for listening.